Good enough. So my name is Paul Whiting. I'm Professor in Institute of Neurology here, and I'm also Chief Scientific Officer of the Alzheimer's Research UK Drug Discovery Institute. Perfect. So I'm going to talk to you about drug discovery and use an example from the neurosciences to go through that. Okay? So, drug discovery is a long process, and that's shown here. And it can take up to 15 years to go from an idea through to a drug at the end of a drug that you can use to treat people. And as you can see, the other thing is that there's a huge attrition rate. So you start off at one end by screening thousands of potential drug compounds, and as you go through, you whittle that down, and at the end, if you're very, very lucky, you might end up with a drug in people. But the problem is, out of that, the failure rate is huge in terms of making drugs. And worst of all, the failure rate for drugs to treat diseases of the central nervous system are the highest risk. And that particular disease area has one of the highest risks of all. So it's extremely challenging. You can see that even once you get a potential drug into the first stages of clinical testing in man, there's still greater than a 90% chance will fail, and that drug won't make it through to be a registered, approved drug. So it's a very costly and difficult approach. I mean, how much it costs to take a drug through from an idea through to patients, that number varies a lot. And it's generally not the cost that's assigned to it, it's not just the cost of that one individual program, but you summate all of the costs in for the 95% of failed attempts into that one success, but it generally works out to be over $1 billion. So it's a very expensive business, all things considered. So we can divide the drug discovery process into two stages: the preclinical and the clinical. So here you can see the preclinical stages, where you come up with an idea based upon animal models or human pathology or human genetics, cell-based models, and out of that you generate a hypothesis and a potential drug target which you want to modulate with compounds. And then you further validate that, you then develop some screens to find compounds, to find potential drugs that modulate the function of that target, that protein, that enzyme. And then tool compounds that you've developed that you can then test again in animal models and cell-based models to test your hypothesis. Do they do what you think they should do? And then after some further optimization of that drug, at that point you can go into clinical trials. The clinical trials have a number of different stages, I'm sure you're aware. So they have phase 1, 2, 3 and 4. So phase 1 is where you're really looking for safety. Is the drug safe? And you're looking for a clue that maybe it does what you want it to do pharmacologically. Then the second phase 2 is then all about efficacy. Can you see in any way an effect on the disease? Then phase 3 is where you go into thousands of patients and you really want to be able to demonstrate that you can statistically see a robust effect on the disease, and you compare that to any other treatments that are currently available if there are. And it's only at that point that you can then present all of the data to the regulatory agencies. In the USA, it's called the FDA, Federal Drug and Food and Drug Administration, and they'll decide whether or not the data is sufficiently compelling that you can register drugs and then you can prescribe it to patients. So that's the way the whole system works, from soup to nuts. So what we're going to do is use as a paradigm, or attempt to use as a paradigm, paradigm anyway, the development of GABA A receptor subtype selective drugs. So I know that you've heard about GABA, or I hope you've heard about GABA in the lectures so far. So GABA, gamma-aminobutyric acid, is the main inhibitory neurotransmitter within the brain. And like all classical neurotransmitter receptor systems, it works in a very standard way. So you have GABA in synaptic vesicles in the presynaptic terminal, and action potential comes along. That leads to calcium influx, and the vesicle fuses with the presynaptic membrane. You get GABA released into the synaptic cleft, which diffuses across and then activates the postsynaptic receptor. So a very classical system. In this case, GABA causes hyperpolarization rather than depolarization of the postsynaptic membrane. Hence, it's an inhibitory neurotransmitter rather than an excitatory neurotransmitter. So as is also always the case, or often the case, there's not a GABA receptor, there's a gene family of GABA receptors. You can see them on the right-hand side here. So there's a whole family. And these receptors are formed from subunits from this family of genes to form a ligand-gated ion channel. And it figuratively looks, as shown here in this diagram, the subunits, generally 2 alphas, 2 betas and a gamma, although there are different flavors, are arranged like staves of a barrel around the central ion channel through which chloride ions flux. So when GABA binds to the GABA A receptor, channel opens, chloride flows through, get hyperpolarization of the postsynaptic membrane. So a very classical mechanism. So the composition of GABA A receptors is thought to be, as shown in, broadly speaking, this pie chart. So these 18 different subunits co-assemble in these various flavors. And there are a lot of them. But actually, once you peel away the non-classical ones here, if I can call them that, then the larger populations can be really narrowed down to a very relatively small number. And these are the ones we'll be discussing. So alpha 1 subunit with a beta 2 and a gamma 2, alpha 2 with a beta and a gamma 2, alpha 3 with a beta and a gamma 2, and alpha 5 with a beta and a gamma 2. So those are the ones we're going to be discussing. They make up the vast proportion of GABA receptors in the mammalian and the human brain. And while we're focusing upon these, as it happens, these are the ones, these are the GABA receptor subtypes that benzodiazepines bind. Benzodiazepines, I'm sure you've heard of them. They were first introduced in the 1960s as drugs. They bind to a specific binding site on the GABA A receptor, which is distinct from the GABA binding site. So a different site. They have no intrinsic activity by themselves. They don't do anything when they bind by themselves in the absence of GABA. But in the presence of GABA, they modulate the GABA response. So they are allosteric modulators for the GABA A receptor. So all clinically prescribed benzodiazepines are positive modulators. So they enhance the activity of the receptor in the presence of GABA. And you can see some structures of very common benzodiazepines at the bottom. They're all variations upon a theme. There's a chlorine or a hydroxyl group shoved on somewhere, but they're all variations upon a theme. So what do benzodiazepines do as prescribed medicines? So what we describe these as non-selective benzodiazepine agonists, i.e. they don't discriminate between these subtypes we're talking about, alpha 1, alpha 2, alpha 3, and alpha 5 containing receptors. So they're non-selective. So they're used as sedatives, they're used as muscle relaxants, they're used as anxiolytics to treat anxiety, and they're used as anticonvulsants. But they have some serious downsides. As I'm sure you're aware, they interact with ethanol. They impair memory. They become even tolerant to their effects. Some of their effects, they induce significant dependence, and hence this gives them a very serious drug abuse liability. What we also know is that there are some benzodiazepines like a common drug called Zolpidem, which are selective alpha-1 containing GABA receptors. And these are very sedative, so they are very hypnotic. So that suggests that certain GABA receptor subtypes may be responsible for certain effects in the body. So the question is, can these various effects of benzodiazepines like this be separated? I.e., are these effects mediated by different GABA receptor subtypes? Because they are, you can think of developing novel subtype selective drugs with improved side effect profile. So you develop a drug to treat anxiety that isn't sedative, or a drug to treat anxiety that doesn't cause abuse potential and dependence. So if you did, you have a really very effective and useful drug to treat people. So what do we know about the benzodiazepine receptor, the benzodiazepine binding site? So we know that the benzodiazepine binding site of the GABA receptor is located in a cleft or a binding site that's made up of the GABA2 subunit and the alpha subunits. And what we know is there's an amino acid called histidine 101, so in the sequence from 1 to 400 or something of all the amino acids in an alpha subunit, histidine position 101 is actually critical to benzodiazepine binding. And we know if we change this, for instance, to an arginine, very similar amino acid, we lose about 1,000-fold in the affinity. So we essentially lose, this receptor then loses its ability to bind benzodiazepines. Now how can we use that? So what we did, or what was done, was to make genetically modified mice whereby the histidine at position 101 in the alpha-1 subunit was mutated to an arginine. And we did this by homologous recombination genetic engineering. So that means that every single GABA receptor alpha-1 subunit in the brain is mutated so that it contains an arginine at this position. So what do these mice look like? So actually, they're unsurprisingly, they're totally normal because there's no endogenous benzodiazepine in the brain that normally binds to this site. So this binding site has no normal function in the absence of a benzodiazepine. And the total number of GABA receptors in these animals was completely normal. And at the bottom in that picture, this is a section through a rat brain where we've added to it a radio-labeled benzodiazepine to localize where the GABA receptors are. And you can see the wild type, there's loads of binding, that's the orange and red, showing all the GABA receptors. And in the mutated mice, the binding decreases greatly. So that's not because the receptors have disappeared, it's because the binding site has lost all its affinity for the benzodiazepine. And when we give diazepam, benzodiazepine to these animals, what happens? Well, it turns out that diazepam in these animals is still anxiolytic and works in terms of treating anxiety behaviors, but it's no longer sedative. So what that tells us is that alpha-1 containing GABA receptors mediate the sedative effects of benzodiazepines, whereas alpha-2 and alpha-3 containing GABA receptors, which in these mice still bind benzodiazepines and still modulated by benzodiazepines, mediate the anxiolytic effects. So this tells us that if we could develop a benzodiazepine which modulated the alpha-2 and alpha-3 containing GABA receptors, but not the alpha-1s, we could develop or have a non-sedating anxiolytic. The other thing, that experiment that was done by people with these animals, looked at the addictive properties, the use liability of benzodiazepines. So one of these people showed, first of all, in the top there, you can see, I hope, is a section through a region of the brain called the ventral tegmental area, which is a region that's full of dopamine containing neurons that is responsible for many of the addictive behaviors that we see in people, in us. And it turns out that this area is rich in GABA-A containing GABA-A receptors. And what these people is they took these mice, the genetically modified mice, and then we asked the question whether they showed any addictive responses to benzodiazepines. And you can see that in the bottom. So the mice have got two bottles presented to them, where the choice where they have either drink water or sucrose or sucrose or benzodiazepine, or a benzodiazepine. And what you can see is when they give them two bottles of water, they don't discriminate between the two, so they've both got water in them. subtype mice dramatically prefer to have the bottle to drink water, to drink the solution that contains the benzodiazepine. Whereas the mutated mice show no discrimination between the two bottles. So they don't recognise there's benzodiazepine in one of them because they're not responding to the addictive properties because they don't have a receptor, an alpha-1 receptor that is responsive to benzodiazepines. So again, that tells us that alpha-1 containing receptors mediate, at least in mice, the addictive properties of benzodiazepines. And these are type of experiments you can do with these genetically modified mice and drugs. So now we've got a hypothesis, we need to move on to thinking about screening compound libraries and developing drugs. So when you think about developing a drug, you need to think about what is it you're trying to do? What do you want your drug to do at the drug target, in this case a GABA receptor? You want to elicit a desired physiological effect, and you want to show efficacy in the disorder or the disease. So to do that, you need to think about what do you need? You need an agonist or an activator? Do you need an antagonist or an inhibitor? Or do you want to have an allosteric mechanism? What is it you want to do? And in this case, we're interested in developing an allosteric mechanism, just like benzodiazepines. Then, in this case, we want to get benzodiazepines or drugs that are selected for particular receptor subtypes. So thinking about that, there are two ways we can go about doing that. So the most obvious one is affinity selectivity. So in this graph here on the vertical axis, we've got a modulation of a GABA response, so the desired effect, and along the bottom we've got the concentration of the compound. And what this cartoon tells us is that for this particular fictional drug, we've got higher affinity at GABA receptor subtype 1 than we do at receptor subtype 2. We can screen for compounds that have these properties, for instance using radioligand binding assay, which I have for selective affinity, which I hope you've heard about already. So we did this, or this was done to try and find compounds with higher affinity at one receptor subtype than at another. What this graph shows, each blue dot is the affinity of a different compound that binds to GABA receptors. And the vertical axis shows the affinity of about 3 containing receptors for all these compounds, and the horizontal axis, the affinities about 1 containing receptors. And you can see they pretty much lie in a straight line, so they have no selectivity really between the two subtypes. So what we've been looking for is compounds in this quadrant here that have higher affinity at alpha 3 compared to alpha 1. So taking an affinity selectivity strategy has not been useful in this case. So therefore we need to think of another way about this. And the other way of approaching selectivity between subtypes is functional selectivity or efficacy selectivity, and that's shown on the right-hand side here. So again, here we've got the desired response, the amount of modulation of the GABA receptor response, and along the bottom we've got the concentration of the compound. And here we can see that the affinities, if you went across here from the 50% effect down to here, the affinity of this particular compound for these two receptor subtypes is pretty much identical. However, one is very, very efficacious against receptors. The drug is very efficacious against receptor subtype 1, but has essentially no efficacy against subtype 2. So this is an alternative way of getting selectivity between different receptors. So this was the approach we took. So, if we're measuring functional selectivity, what types of assays do we need to measure the effect at ligand-gated ion channels like a GABA receptor? The gold standard is electrophysiology, which I'm sure you've heard about. And over a period of years, this has changed from looking literally at slices through brains to measure the effects of drugs on receptors, through to single patch clamp electrophysiology, looking at the effects in culture of single cells, through to high throughput electrophysiology. Electrophysiology is very high fidelity, but until these new technologies, it's very low throughput. There's also non-electrophysiological assays to measure the effects, to measure the function of ion channels. And we used to do this by a very horrendous method, which is using radiolabeled chloride and literally measuring the amount of radiolabeled chloride that's taken up into the cell when you put GABA onto cells containing GABA receptors. And it's pretty horrendous stuff. This is a pretty horrible radioisotope. More recently, there's been fluorescence, voltage-sensitive fluorescent dyes that can be used to measure the voltage change of the cell membrane in response, for instance, to GABA receptors opening in response to GABA. And now there's a whole bunch of very expensive equipment that you can use to screen for drugs that act on ion channels. And these are high throughput electrophysiology pieces of equipment. And they range, and here we've got something like manual patch electrode, which is someone doing electrophysiology manually, which can screen only 1 to 2 drugs, 1 to 2 compounds a day, so very low throughput, all the way through to some pieces of equipment with a high throughput electrophysiology that can screen thousands of compounds a day. So if you're doing quote unquote proper drug discovery for ion channels, this is the kind of equipment that you have to use. So we took this approach to screening to try and identify drugs or compounds that selectively modulated GABA receptor subtypes. And we found this particular drug here called TPA-023. This is its structure. And it's quite different from classical benzodiazepine diazepam. And this trace here shows the effect of TPA-023 on recombinant human GABA receptors expressed in mammalian cell lines. So here we've got an inward response. This is the current in response to GABA, at alpha-1 containing GABA receptors, and this is the response to GABA in the presence of this drug, this compound, and you can see no effect at alpha-1. When we do the same experiment at alpha-3 containing receptors, this is GABA, and then GABA plus this drug, you can see an enhancement. So a positive allosteric modulation of alpha-3 containing GABA receptors by this particular drug. And you can see this graphically over here, that this drug, this compound, allosterically enhances the GABA response at alpha-3 and alpha-2 containing receptors, but very little at alpha-1, at alpha-5, and nothing at alpha-1. So this was the type of drug compound we were trying to develop. But what does it do when we test it in animal models? So we move on then to taking the tool compound and looking in animal models. And so there's some very some might say weird animal models that people use to try and determine their behavioural and physiological effects. And one test that's used to determine anxiety is called the elevated plus maze, and that's shown on this figure here. So here we've got a plus maze, you can see it's got two arms. This is the open arm going across here, and this is the closed arm. And generally what happens is that when it's shone by a bright light, and generally what happens is animals, particularly rodents, don't like this. It's off the ground about a metre, and it's bright light, and they prefer, being out here is quite anxious for them, they prefer to be in these closed arms. And so you can see here, they spend most of their time, just when given a vehicle, so the placebo, within the least amount of their time is on the open arms. When you give them a dose of this drug, this novel anxiolytic, you can see that the amount of time that they spend on the open arms increases. So that is interpreted as an anxiolytic and anxiety effect. And this is a positive control of low dose of benzodiazepine. So that suggests this drug is anxiolytic. So the other question is it sedative? So here's an even more Byzantine animal model that's used to measure sedation. It's a very sensitive measure, apparently, and it's called, hilariously enough, the rat chain pulling model. And so what happens here, we've got a chamber here, I think you can see there's a chain there, and when the rodent pulls this chain, it releases a food pellet down here that the animal releases. And animals, rodents, will continue to feed themselves by pulling this chain once they've learned this task. And you can see that here animals are just given a vehicle, so nothing really, when they're given diazepam, you can see their response is very, very low. This is primarily because they're sedated. They're basically almost asleep at the bottom of the chamber here. When they're given this non-sedating anxiolytic, TP023, you can see that their responses are pretty much the same as the vehicle. So they're not sedated in this animal model. So again, that is data to suggest that this drug, TPA023, which is selected for alpha-2 and alpha-3 containing GABA receptors, is not sedated. The other experiment that was done in this, with this particular drug was done in an animal model, which is a squirrel monkey conditioned emotional response model. So in this particular model of anxiety, animals are trained to press a lever for a drink reward. And then they're trained to understand that when a red light randomly comes on, the so-called conditioned suppression, this will suppress, which precedes a very, very mild electric shock. This suppresses their drinking, they're obtaining the drink reward. So here you can see animals given vehicle pretty much are suppressed when the red light goes on, they don't drink. Whereas animals given the anxiolytic in a dose-responsive manner, their willingness to drink when the red light goes on goes up because their anxiety is removed. So again, this is consistent with an anxiolytic effect of this drug. So now we've got a compound that does what we want it to do. We need to move forward into thinking about how to progress it in patients. But one important test that has to be done, in fact, the regulatory agencies like the FDA actually require pharmaceutical companies to do this test, is to test abuse liability. It's considered a really important test to do. And so this is the test that drug companies, pharmaceutical companies are required to do. And what it is in this case, it's a baboon that's hooked up intravenously to a lead that enables it to self-administer the drug that happens to be in the chamber here. And in this case, it's either a standard benzodiazepine called diazepam or this drug. And you ask the question, what's the number of injections over a period of days that the baboon gives itself? And I think what you can see here is that when it's given access to diazepam, the number of injections that it gives itself goes up, and that goes up in a dose-responsive way. So clearly, diazepam is a drug of abuse, and we know that. So that's a positive control in this experiment. However, when it's given this novel drug, TPA023, you can see there's no change, which is consistent with it not being a drug of abuse. Now, one argument you could say when you look at this experiment, you could say, well, that's all very well, but how do you know the drug gets into the brain? Maybe the drug doesn't get into the brain of baboons, so therefore it's not going to do anything to the GABA receptors. So you have to show that the drug gets into the brain. In fact, you have to do this with people as well. It's an important experiment. So the way we do that, or the way that's done, is through PET, positron emission tomography. This is a way of demonstrating the drug gets into the brain and binds to its drug target. That's a PET scanner. And PET works, as I'm sure you'll know, by having an isotope in your compound of interest, in this case a drug that binds to benzodiazepine receptors called flumazenil, having an isotope C11 that is a positron emitter. And when that's in there, positron decays to emit to electrons and photons, and the photons can be detected by the camera, and you can localize to quite high resolution where that signal is coming from in the brain. When you do that, here we're giving, in this case, the monkey, I think the monkey, I can't remember, a human, it doesn't matter, this is just an example, where they've been given C11 flumazenil, and you can see the nice signal which literally localizes where the GABA receptors are in the brain. So it's a really cool system. And here you can do pharmacology in it. So here, the person or the individual has then been given a benzodiazepine. And you can see the signal goes down by 64%, and then over time goes up as the drug disappears. The inhibition is now 34%. So that's a radioligand binding assay. We're competing off the C11 flumazenil for binding to GABA receptor by the other drug that we've given to the individual. So it gives a direct measure, a quantitative measure of how much of the drug we're giving gets into the brain. And this was done in these baboons. And what you can see here, this is a baseline showing in the baboon, given TPA, given just placebo, those are finding a C11 imaginal with GABA receptors in its brain. Then in a dose-responsive manner, as we give increasing amounts of TPA-023, you can see that the signal for the PET ligand goes down because it's being competed off by the drug. And you can see by the time we get to 0.32 meters per kilogram of drug, there is absolutely no binding effect. So we've got 100% occupancy of the GABA receptor by our drug. And that dose, if we go back to this, is a dose that shows no abuse liability. So we know that when we're giving enough drug to occupy, to bind to all the GABA receptors in the brain, that's a dose that still doesn't evoke any liability. So that's a really nice effect. So putting this all together, this technique allows you to integrate the amount of receptor in the brain being bound by the drug, by the dose, if you will, that you're giving, the amount of drug in the blood, and by how much of that dose and occupancy at the receptor in the brain evokes particular effects. So an anxiolytic effect was evoked by 65% occupancy, and then didn't see any sedation or any abuse liability even at the highest doses. So it allows you to integrate. So it allows you to understand you need to give this amount of drug to this patient to evoke this response. So it's a really important measure. So what happened in clinical trials with this drug? So what happened was that it turned out that TPA-023 was significantly less sedative in people than non-selective benzodiazepines. So that part of the hypothesis was correct. But unfortunately, as often happens with drug development, it was found that this compound had significant adverse effects. It caused cataracts in the lens, and so therefore its development, because of safety reasons, was not progressed any further. So the key experiment in terms of anxiety was not really done. It was also taken forward into a clinical study in schizophrenia to look for any effects on cognitive impairment, in fact working memory. And the hypothesis, which I won't go through now, which you can read if you like in slides on Google, is shown there. But unfortunately, yet again, it was a nice hypothesis, but it didn't work. So that's as far, unfortunately, as that drug actually got in development, which sort of plays back to my initial comment that the less than 1 in 12 drugs that makes it through to clinical studies actually progress through to making it to a drug. But what about other GABA receptor types in the brain as potential targets? So as I said, one of the main side effects, if you will, of benzodiazepines is on memory. Non-selective benzodiazepines like diazepam impair spatial learning in rats. They're amnesic in people, which is an unwanted side effect. And although it is a useful, actually, effect in surgery patients, and people are often given benzodiazepines not just because of their myorelaxant anxiety, but because they impair memory. So if you undergo a procedure, you probably won't remember exactly what it is when you wake up on the other side of the procedure. There are non-selective benzodiazepine inverse agonists. So what inverse agonists do is rather than potentiate the effect of GABA, they actually inhibit the effect of GABA. And as it happens, when you give non-selective benzodiazepine inverse agonists to animals, they are pro-cognitive. They enhance spatial learning, but because they're non-selective and they are essentially inhibiting the function of all GABA receptors in the brain, they're actually anxiogenic. They cause anxiety, and they are pro-convulsant. They cause they enhance the potential to have seizures. So they're not a good thing. But the question becomes, would a receptor subtype selective inverse agonist be cognitive enhancing and lack the anxiogenic and pro-convulsant effect of non-selective compounds? And if so, would this be useful, for instance, in Alzheimer's disease or Down syndrome or dementia or whatever? So, if we look at one particular receptor subtype, that's the alpha-5 containing GABA receptor subtype, This is actually a pretty small slice of the whole pie in terms of GABA receptors in the whole brain. But actually, if you look at where it's localized across the brain, you can see that it's in the rat brain here, and also in the human brain here. It's very much localized to a region of the brain called the hippocampus. And the hippocampus is a region that I'm sure you've heard of. It's very important in cognitive function, particularly in memory, in laying down memory. So to evaluate that further, we made a mouse, an alpha-5 GABA receptor knockout mouse. So this is a mouse that has had the alpha-5 subunit deleted using genetic engineering. These animals are apparently normal, healthy, reproduce normally, but they have no alpha-5 GABA receptors in their brain. You can see that here, sectioned through wild normal and knockout mouse using a radio label. It happens to bind selectively with GABA receptors. Here you can see it bind really nicely to the hippocampus, the structure here, and it's completely gone in the alpha-5 knockout. So we know that we've knocked out the mouse. So what does it do to the mouse in terms of behavior? So again, to test that, we use a very interesting animal model. And this is called the Morris water maze, after someone called Brian Morris, who is now a professor of neuroscience at University of Edinburgh. And as it happens, for anyone who listens to Radio 4, and I'm sure it's essentially nobody, but he was on the Life Scientific, which is some Radio 4 show they have on, I think on Tuesdays, I don't remember, this week. He was the person who was interviewed about his life in science, and he's really into cognition. So he invented this test. So this test consists of a pool, a tank of opaque white water. See it there. And you can see a rat swimming around in it. And somewhere located in this is a submerged platform. And what you put the rat in, the rats would prefer not to be swimming around in the water, so they swim around, and the first time in there, eventually they'll bump into the submerged platform and they'll sit on it. And you can see the rat here sitting on a submerged platform. What the rat then does is it looks at these spatial cues around the wall, and it sort of computes where it is. And so ideally, the next time that it sits, it goes on there, it's shoved into the tank. It will remember where the submerged platform is and swim there more quickly to get out of the water. And that's shown here. So here we're looking down from above, if you will. We can see the rat was shoved into the pool somewhere, I can't see anywhere, somewhere it was dumped in here. And then it swam around, and finally found the platform. It took 52 seconds. Second time it went in, it took 21 seconds. So there was a savings of 31 seconds because it remembered where the platform was. So that's its spatial learning. So what do the knockout mice do? As it happens, the alpha-5 knockout mice perform significantly better in the water maze. So the wild-type mice had a savings between the first time and the second time they went in the pool of about 3 seconds. The knockout mice had a saving of about 9 seconds. So they performed much better in this particular test, and that showed graphically over a series of trials here. And it wasn't because they were swimming faster, it was because they took a shorter route, because they could remember where the hidden platform was. So if a knockout mouse does this, can we make a drug that is an inhibitor and an antagonist, an inverse agonist of alpha-5 containing GABA receptors, and would that do the same thing? So we again took the same route as screening compounds for finding drugs that selectively inhibited alpha-5 containing GABA receptor function. And one drug or compound that did this is alpha-5IA, shown here. And here's its effect on alpha-5 containing GABA receptors by electrophysiology. This is the inward response to a dose of GABA. And here's the response in the presence of the alpha-5 inverse agonist. You can see it's inhibited. And that's plotted out graphically here. It's a very potent inhibitor of alpha-5 containing GABA receptors, but much less at the other GABA receptor containing subtypes. So what does this do when we dose mice, normal mice, with this compound and test them in the Morris water maze? And you can see here again, in a dose-responsive manner, when given this drug, the mice have increased savings in terms of their ability to remember where the hidden platform is in the Morrison automaze. So this drug enhances their performance in the Morrison automaze, enhances their spatial memory. So this drug then went into people to look at the effects in so-called cognitive drug research batteries. So this would be one of those phase 1 studies. And the test was, after he's done a safety study, went into this experimental study in normal people, normal young, healthy, and normal elderly. And they were given the drug, and then they were given a computerized cognitive test battery to analyze a number of different parameters, such as their reaction time, vigilance, their spatial memory, their working memory. And they were dosed for 2 weeks, and they were tested across that period of time. And unfortunately, the drug didn't do anything. It had no effect on the performance ability of either healthy young or healthy old. It was a real shame. And this was given at a drug dose that was predicted to occupy 50% of the receptors. It's not clear why that was. Is it that 50% occupancy of alpha 5 receptors is not enough? Or is it because you need to go higher dose than that? Or is it because actually it's just a rubbish idea and actually mice models are just not predictive of human behavior? Don't know the answer to that. But the drug was also given in another experimental medicine study to see the effect on alcohol-induced memory impairment. And so this was an experiment that was done where in young, healthy males, social drinkers, where they were given the drug, 4 milligrams or placebo at time 0, and then minutes afterwards they were given, about 2 hours afterwards, they were given some alcohol, and then they were asked to learn a list of words and then asked to recall the words. And what you can see here is that alpha 5, those when they're given the drug, the inverse agonist generally improved recalled words relative to placebo, and there was no adverse effect here. So there does seem to be some biological effect on attenuating alcohol-induced memory in man. So this is an experimental paradigm. What this means in terms of treating diseases is unclear. Another experiment that was done was to give this drug to animal models of Down syndrome. So this is a mouse model called TS65, which is trisomic for orthologs of about half the genes on human chromosome 21, which I'm sure you know is trisomic in Down syndrome. So it's a mouse model of Down syndrome. And again, in this animal model, alpha-5-IA, the compound given to these animals, restored learning in these animals. So this is much like the Morrison automaze again. This is an improvement in learning, in spatial learning over time when they're given the drug. So this is the impaired animal, the Down syndrome model, which is a lot slower to learn the test compared to the field blocks here. The trisomy 21 Down syndrome models given the drug. You see a big difference. So again, in this animal model of Down syndrome, the drug seems to be working to improve spatial memory. So this drug was actually given to individuals, Down syndrome individuals. This was done, I think, a couple of years ago, 2 years ago, in an efficacy to see whether there was any effect on cognitive function and memory in individuals with Down syndrome. And unfortunately, this trial read out in July this year, and it did nothing in individuals, adults, or either adolescents with Down syndrome in a very well-controlled study. So, what does this tell us? It tells us it didn't work, but what does it tell us? I think it tells us that we need to rethink the way we go about drug discovery. I think it certainly tells us that our animal models The other thing that I think is very important now is really using human genetics to inform our selection of drug targets. That's really, really important. And now with the advent of whole genome sequencing and really getting a much deeper understanding of raw genetics in human disease to identify pathways that are being affected in disease and individual drug targets within those pathways, that's definitely the path forward. And then the final path forward is experimental medicine paradigms. So just like that experiment, the alcohol attenuating the effects of alcohol in young healthy males given the drug, taking these kind of experimental medicine approaches to test our hypothesis very early on in drug development means that we can try and do key experiments before we have to go into a large phase 2 clinical study which costs tens of millions of pounds. So the idea will be to test our hypothesis very early and fail early. So that's really all I wanted to say.